This is Chaitanya. Today I will be presenting our project status. So our project is DNA compression for efficient storage and analysis. So in this project, we want to implement various optimized compression algorithms, both lossy and lossless. So lossy algorithms are the algorithms which lose certain amount of information permanently to achieve higher compression ratios, and lossless algorithms don't lose any information. So we have our primary aim here is to compare methods between different trade-offs uh, for minimizing the data size as much as possible while preventing the essential information. These are the different contents in the PPT: abstract, introduction, objective, background, methods, and current status. There has been a rapid growth of genomic data in the recent years. So there is a need for efficient compression techniques to store these sequences. DNA data sets have a unique challenge because of their size and structure. Also, they require domain-specific optimizations. What it means is that uh, since the DNA datasets have only ATGC and they have certain patterns, repetitions, and structures, so traditional algorithms may not be that effective on these datasets. So our objective here is to implement various compression algorithms, both lossy and lossless. So we want high compression ratio, fast compression and decompression, and accuracy and control loss. So there shouldn't be any uh, loss of information while compressing and decompressing the data set. So we'll be analyzing certain trade-offs among the different methods uh, and comparing them, which include compression efficiency, computational resources, and data fidelity. Many compression algorithms have been developed uh, for compressing large data sets, uh, but these algorithms will not work that effectively against DNA datasets. As I said before, DNA datasets have certain specific patterns, such as the reference-based structure. So even in the developed algorithms for DNA datasets, there are challenges like balancing compression ratios, speed, and scalability for large datasets. So these are the different methods which I will be implementing for this project. Context-based arithmetic coding compression, run length coding and Huffman coding, uh, relative impulsive and approximate matching based compression. So this is a context-based arithmetic compression. It is a lossless technique. So it basically predicts the probability of a certain nucleotide to occur, and it adapts dynamically to the data, and it gives shorter codes to the more frequently appearing symbols. This compression technique can achieve a high compression ratio, but is a bit computationally expensive and complex to implement as well. There's run length coding and Huffman coding. Both are separate alga techniques, but they can be combined to get good results. Uh, they are lossless techniques. So RLE will replace repeating characters with that character and its count. So if you have A, it will replace it with 3A. And the output which we get from the run length coding, we can pass it to the Huffman tree, which creates the Huffman code table, and in which the frequency of the symbols are converted into binary, and it is converted in such a way that The shorter binary codes are given to more repeating symbols, and longer binary codes are given to less repeating symbols. We'll see this in detail in the upcoming slides. This is relative impulsive compression. It is also a lossless technique. Here we are using a reference sequence. So there are two ways to do this, intra-referencing and inter-referencing. For intra-referencing, we use a portion of the same DNA dataset uh, as a reference to compress the rest of the sequence. And for inter-referencing, we use a DNA dataset that is related to the dataset we are using, or that is similar in structure to the dataset we are using. So instead of storing the entire data, which increases the space, we only store the symbols that are not in the reference sequence and in the data set, and we'll point to the reference. So this decreases a lot of space. But these algorithms uh, may not work that effect. These algorithms may not work that effectively against very large data sets. So that is a limitation. This is approximate uh, matching based compression. This is a lossy technique. So there is loss of information while decompressing, which cannot be retrieved again. So here also we use a reference, but uh, even if our data set we are using in the reference sequence don't match exactly, it will still uh, compress the data. So this causes loss of certain information. So it makes it a lossy technique. So I have implemented a run length coding and Huffman coding. I have integrated both of them in decompression as well. So the size of the file I used was 161 MB. I used the extracted 23rd chromosome of human chromosome dataset. Uh, and the compressed file size was 44 MB with a compression ratio 3.63. So in this PPT, we have explored various compression techniques and uh, we have explored various compression techniques, the different trade-offs to compare between the compression, the need for compression, and the background. Uh, and for the current status, I have implemented run length coding and Huffman coding. So next, we'll see the code for the run length and Huffman coding compression. So this is the code for run length encoding and Huffman coding. So I'll first give an overview of how it works. So basically, first we pass the data set into we'll call the run length encoding method. So if we have A, it will get converted into A3. So it takes care of that. So this method basically takes care of that. Once we get that, we'll create a frequency table which will have, for example, A3. If A3 repeats three times, the frequency table will have A3 mapped to 3. And then we'll build the Huffman tree. So basically, how a Huffman tree is, it has nodes and it's a binary tree and it has nodes and two children. So the node will have symbol along with its frequency. So the way we construct a Huffman tree is we'll implement a priority queue using min heap, and basically we'll remove the two nodes with the lowest frequency from the queue, and we'll combine them into another node, into single node, and then we'll add this node back to the queue, and we repeat this process until there is only one node left. and that node will be the parent node for the Huffman tree. And then we'll build a Huffman code table, which will have the nodes mapped to which will have the symbol mapped to its frequency. And this frequency is in the form of binary, which is zeros and ones. Then the Huffman encoding is done. So once we, so this is related to the compression. And for compression, basically in compress FASTA method, since the DNA data set since the DNA data set is very big, so we'll pass uh, it in chunks uh, to make the process faster. So the compress faster method will call the compress a block method for every chunk. And here we'll call the run length encoding. We'll build the frequency table. Build a Huffman tree, Huffman code table, and encode. This is everything related to how compression works. Now, when it comes to the decompression, a decompressed file has metadata and encoded data. So metadata has some information about how the encoded data is. For example, the first 4 bytes of the metadata will have is a block header. It will have the length of the encoded data. The next 2 bytes will have the number of unique symbols which are there in the encoded data and more. Then we'll call the Huffman decode method. And in Huffman decode method, we'll basically uh, a reverse map from the binary data to the symbol. We'll get the symbol from the binary data, and that is a Huffman decode method. From there, we'll call the uh, run length decode method. So once we get the symbol from the Huffman decode, we'll get the run length. We'll run the run length decoding method. What it basically does is if we get A3 after the Huffman decode, it will again convert it into AAA. So we'll again get the original data. So in this way, the compression is done. And the result we got was the compression ratio we got was 3.63.